You are listening to Neurosalience, the OHBM podcast.
Welcome to the Organization for Human Brain Mapping Neurosalience podcast. I'm your host, Peter Berrettini. Here I interview neuroscientists and discuss their work, as well as latest developments, issues, and controversies in the field of brain mapping. And today I have a truly inspiring chat with one of the OHBM 2021 keynote lecturers, Professor Nick Weiskopf. He's the director of the Department of Neurophysics at the Max Planck Institute for Human Cognitive and Brain Sciences in Leipzig. He received his Ph.D. in 2004 from the Graduate School of Neural and Behavioural Sciences and International Max Planck Research School in Tübingen, Germany. And also in 2004, after he received his PhD, he moved to the Wellcome Trust Center for Neuroimaging at University College London, first as a senior research fellow, but quickly moving up to head the physics group. Finally, in 2014, he became full professor of MRI physics. However, in 2015, he was recruited to this directorship position, the directorship of the Neurophysics department at the Max Planck Institute for Human Cognitive and Brain Sciences. And this position we talk a little bit about the uniqueness of this position and the uniqueness of the Max Planck Society in general. So regarding the Department of Neurophysics, the vision is to develop and apply functional microstructure imaging and in vivo histology using magnetic resonance imaging as novel, non-invasive MRI methods to reliably characterize detailed functional and anatomical microstructure of the human brain. And in this regard, he has been overwhelmingly successful so far, producing consistently pioneering work and defining and surpassing the limits of what structural and physiologic information MRI may derive from the brain. This was a unique podcast in that while I knew his work, I didn't really know him personally. And I realized that in a way we were kindred spirits. We're both physicists working at the interface of MRI acquisition and brain physiology and function. And we're trying our best to plumb ever more information, quantitative information, about how our brains are organized and how they change with disease and how they vary among individuals. We talk about his professional history, get into some talk about his past work with real-time neurofeedback MRI, and then delve into his work on quantitative MRI for deriving maps of myelin, iron content, fiber tract direction, and more, all at unprecedented resolutions using their unique Connectome scanner, which is one of the only 4 in the world so far. So I hope you enjoy this podcast. Nikolaus Weiskopf, thanks for coming on to this podcast. And I've been following your work for years, and it's nicely straddled between basic MR physics fundamentals in MR physics and acquisition, all the way to neuroscience. So just to begin, how did you get started in this field? What got you? I mean, somewhere along the line, what sort of piqued your interest in terms of using MRI and using it as well as possible to extract functional information or physiologic information?
Right. Yeah. Thanks for the very kind introduction, Peter. It's great pleasure and honor to be on the podcast. And yes, I mean, how did I get started with MRI? Basically, I studied physics as my main topic. And then I was looking for topics where you are quite close. First of all, you are studying something interesting, and it was very clear to me that the brain is something truly, truly interesting. We need to put in a lot of thought and work to understand it. So that was clear. And I also wanted to work in an area where you get results rather quickly, where you're not so far away from results. And as we know in physics, you have sometimes these huge experiments like in CERN or in other places where you really have to work for years before you get the first results. And when I saw kind of in general the neuroscience area domain, neuroimaging domain, I thought that that's a really interesting area because you can get quickly results. You can really advance understanding still rather quickly because I think it's still relatively early science. So I really liked working there, and as a physicist, I had the feeling I can really bring some skills, some knowledge to the table. And incidentally, I actually didn't start with MRI. I started with magnetoencephalography, with MEG at the time, and working in a rather exotic topic. It was measuring DC fields, so very slowly varying fields at the time, and this had to do actually with my latest supervisor then, because Niels Bierbaumer in Tübingen, who worked a lot on brain-computer interfacing. And at the time, they were based on slowly varying fields, for example, slow cortical potentials, SCPs. And so there was a general interest in the community there. And then after I'd worked with MEG, I already during that period of time, I got in touch with a lot of colleagues who actually worked in the MRI domain in Tübingen. It's a big place. A lot of people like Wolfgang Roth, for example, at the time had already done functional MRI for a long time. And I got really fascinated by that. And then basically Niels Bierbaumer approached me at one point and said, look, you're a physicist, you are the smart guys who can program these scanners and work with those. And I have this long-standing problem that I'm working with these brain-computer interfaces, with neurofeedback. And the problem is with the EEG, and they were EEG-based, the problem is we can't really see deep inside the brain, the deep brain areas. And Niels was very much interested in the amygdala and other deeper brain areas. They were incredibly hard to sense with EEG at the time. So he had this idea. He had heard about real-time FMRI. Maybe one could build a brain-computer interface with that and do neurofeedback with that. And that's how I came in, because then I thought, okay, this is interesting because on the one hand, I'm going to learn about the brain. On the other hand, I'm actually going to apply my skills as a physicist really well. And I think I was extremely lucky at the time that I had a great thesis advisory committee with Niels Bierbaumer, with Klaus Mattiak and Uwe Klose. So a nice mixture of physicians, psychologists, physicists, also in this advisory board, thesis advisory committee.
And did you overlap? I guess when you were in Tübingen, it's I've been there twice. Logothetis was there at least, and I don't know if that was it's a great center, and certainly they have the and I didn't know that you were actually started in MEG as well. That's interesting. And I noticed actually you've done a little bit of MEG just recently, trying to look at the correlation. Fiber orientation and iron content with the MEG signal. But so as far as now, and I recall, obviously, Bob Turner was in charge of the director of the Department of Neurophysics at the Max Planck Institute in Leipzig, and he retired. Although luckily not completely, he's still sort of active. And I was sort of aware of that position opening, and it's a big deal. And I actually was impressed with. It's not just hiring someone to be in the States, for instance, being a professor or assistant professor or whatever. It's like it's the Max Planck. I like to think is something like the NIH, where you have permanent positions and you have hard money and you have a group, but in this particular position, it actually is it's more significant. Do you want to talk about that a little bit, your position and what it entails?
Yeah, sure. As you said, I think kind of working in the Max Planck Society in general, but in particular, of course, when you direct one of the departments, I think it is very special indeed in terms of I think one of the things that is special, that it's a society that is targeting basic research, fundamental research. So, you can really dig deep, you can try to kind of crack really the hard problems, and you're given the time and the resources to actually do that. So I think that's rather special, and that applies to, I would say, every level in the Max Planck Society. And what it also means is that typically also have pretty unique facilities. So I think we are very lucky here. We have, for example, a 7-Tesla MRI scanner to do ultra high resolution functional MRI. For example, the laminar FMRI, as you do it as well. And we also have one of the 4 Connectome MRI systems. So we can do with a very strong gradient system. So we can get excellent diffusion imaging, for example, acquired here. But I think what counts more than the facilities is really that mindset you can rely on, and you get support from everybody. And I would say even from the administrative groups, you get this support. They ask you questions about how they can support your science and how they can kind of speed it up or expedite it. So I think that's a really, really special place to be.
Yeah, no, that's and I agree with you 100%. It seems that there's pluses and minuses to each system, but certainly the right now the system of the extramural world, at least in the United States and elsewhere in Europe as well, is people are scrambling for grants, and they're constantly writing grants, which does, I think, keep people sharp in terms of always trying to find the most interesting thing to get funded. Most of the time they're not funded, and sometimes they go for short-term problems in some regard too. So there's always these trade-offs as far as that system at the NIH and Max Planck, and it seems definitely at Max Planck is where you can you're given the resources, and they only give this to people that are extremely motivated and extremely focused on the problem, and I think that really does help in many ways, having this stability and the resources to sort of step back and take a deep breath and actually think carefully in terms of what to do. So that's great. So what was it like? I know Bob Turner sort of was there initially. He may have coined the term neurophysics, which I really like. But what was it like? How have you changed the department, or kept it the same after Bob left?
It's a good question. I mean, in terms of the philosophy of the Max Planck Society, when a director leaves, so for example, you're emeritus director, you retire, then they close the department, and that was the case here as well in Leipzig. So the department was closed. So I basically came in and opened it up again, turned the key, opened it up again. And there was quite, what comes with it is obviously the typical, starting, ramping up the activities aspects. At the time, what came in at the same time was, for example, the Connectome MRI scanner. So that was kind of significant amount of work to really bring this up and running and make the best use of it, for example. So that was certainly a new aspect coming in terms of the facilities. In terms of the department itself, I think one of the things I kind of try to really take on board in terms of the mission of the department, the approach is kind of the idea that you pursue kind of 3 pillars there, three directions, and one of them is this classic, let's say, MR physics approach. You try to maybe improve the signal to noise by going to higher fields. You work with new RF coils. You create new pulse sequences, image reconstruction, all these kind of things. On the other hand, we really took to heart and very seriously the biophysical modeling. So this is one of the kind of really important foundations of our work now. And I should actually just mention briefly what one of the big goals is of our work is to identify microstructure non-invasively, to do something like in vivo histology, get information about microstructure similar to histology, but non-invasively using MRI. And for that, the biophysical models play a big role because they help us interpret the data, the image processing. So basically all the computing. So I've ramped up the computing actually quite a bit. We have also close collaboration with the high performance computing center of the Max Planck Society in Munich. So we are in Leipzig, and there's another part in Munich, for example, and they have these big computers basically for processing, simulation. And the last part, the third part, is really doing the neuroscience, kind of these proof of concept studies, smaller, experimental studies to make a point, to validate things, and also postmortem studies for validation. So I think what is kind of what I see kind of the unique package is bringing these three things really closely together. And very often this means there are persons who work on more than one of those areas. So they work in between, interdisciplinary, and we have a lot of external collaborators. And I think this is kind of a special flavor when you ask, what makes you, what's an aspect of your department that is maybe different from other places or other sites in that regard. And maybe I should also mention this when I said microstructure imaging. Overall, what we are obviously very interested in is this entire loop of how function relates to structure, to microstructure, to behavior. We are interested in that in the entire institute, but also in our department. And so we also do a lot of ultra high resolution functional imaging, for example, looking at columns, at layers, and so on.
Yeah, that's a wonderful setup. And I think that you've, right, over the last few years, just getting that started. Yeah, it's really nice. The nice thing about MRI is that it's not just sort of this technique that you press a button and get whatever image you get. There's so many parameters that you can adjust, and there's so many opportunities for potential clinical applications or understanding the brain in a fundamental way that makes it so rich. You mentioned your Connectome scanner, and I think that it's important to emphasize how unique that is as well. I think I think there's only 3 or at the most 3 or 4 maybe in the world.
4 altogether now. Yeah.
Okay, and maybe just describe a little bit how because your images actually that we'll talk about are spectacular, and how that really does allow you to do a lot more.
Sure, I'd love to. I think it's a fantastic scanner. I mean, and in particular for diffusion imaging, and probably a lot of people will know, but what you need for diffusion sensitization and encoding in MRI, you need gradients. You need strong gradients ideally, because that makes the time you need for diffusion encoding shorter. The stronger the gradients, the shorter the encoding time, and you still have basically the same diffusion weighting. And that's how we kind of use it mainly at our site, is basically playing out these very strong gradients. And to give you a number, I mean, the typical standard scanners, they are now somewhere between, let's say, 40 to 80 millitesla per meter gradient amplitude. The Connectome has a 300 millitesla per meter gradient amplitude. And the effect of the gradient amplitude on the diffusion weighting goes in with the square of the amplitude. So you really gain a lot of diffusion weighting. And shortening that diffusion sensitization time means, for example, you have much shorter echo times in your MR acquisitions. And as we know, when you shorten the echo times, the signal goes up. You have much more signal left, signal to noise is higher. And since most of our MRI techniques are actually SNR, signal to noise limited, what it means, we can immediately trade it off for resolution, for example. We can make the voxel sizes smaller and resolve more interesting structures. And that's probably one direction where we maybe now not differ so much anymore from other groups, but initially we differed quite a lot from other groups in a sense that we were interested in diffusion in the cortex and just underneath the cortex. So like the superficial white matter, U fibers, and also intracortical diffusion features. And we use that high resolution to actually be able to get meaningful images, which you can analyze at that level. And for that, you need about at least 1 millimeter resolution, isotropic resolution or higher. And So we are now at 800 micrometer isotropic resolution, which we use for our kind of proof of concept neuroscience studies, for example.
That's actually another thing about MRI too, is that with the methods keep on, I mean, it's been, the field is almost, 40 years old, and the methods keep on improving with the engineering, and then that fosters new physics insights and new physiology insights and new techniques as well. So it keeps on growing as far as that's concerned. And definitely one of the areas where the cutting edge is these Connectome scanners that, we're hoping to eventually get something like that as well. So before we get into what you're talking about at OHBM, for the most part, I just want to talk a little bit. I first became aware of your work in the context of sort of real-time fMRI and the idea of using fMRI, real-time fMRI, where you process the functional images in real time and then feed it back to the subject to allow for training or other types of feedback as well. And you had some really nice papers. You had a Nature Reviews Neuroscience paper in 2016, so it's not that long ago. You've collaborated with a number of people, and you did this even when you were at UCL's as well, I think, for the most part. And just even recently, you have a real-time decoding of covert attention visual areas. So my feeling about real-time is that it's obviously really interesting, and people would love to use it clinically as like a therapy tool. I worry a little bit about because you're relying on a hemodynamic response, that delay. I worry a little bit that it's almost too long to actually foster to be as efficient as, for instance, MEG or that sort of thing. So I'm actually curious about your take on real-time fMRI, and if you're doing it or if you're still thinking about it, and what you think the limits are or the ultimate applications of it might be. Sort of a loaded question, but
yeah. quite a few things maybe to unpack there, and certainly also I would say probably not without controversy in the community and field. There are different opinions on it. Maybe coming back to your comment on the HRF and the fact that we are working with indirect measurements of brain activity in this case, and a bit sluggish the responses. And so the anecdote would be that when we started the neurofeedback work, we were actually rather surprised. So using the bold signal, we were rather surprised it works rather well. And because exactly of that, because before that, Niels did all the EEG neurofeedback work, and all that was established, and there were all kind of paradigms, and people believed that you need this fast response, you need to measure that. And I think this is still not completely clear, but I would probably think there are a few reasons for it. One is maybe signal to noise again, that signal to noise can be rather good in bold imaging, especially when you look at larger regions of interest. And that really helps people to learn when they get reliable feedback, for example. That's not necessarily always the case, at least at that time with EEG. I mean, this has also, of course, made big advances now in the meantime.
I definitely have to add, definitely the fidelity of the signal is a strong point with bold relative to MEG or EEG. It's a little bit, like you said, it's a little bit more, it's getting better, but that's a key advantage. Yeah. So come on, sorry.
Yeah, sure. No, of course. And the other aspect, and it's kind of related to this fact that you have this slow responses, that you would run different types of paradigms, and you would probably target different kinds of, let's say, cognitive processes, perhaps, that are slower intrinsically and that don't really benefit so much from the high temporal resolution. So when you think about, for example, colleagues who have done work on emotion induction or something like that, it even turned out, in practice, that you were almost too fast, that people had trouble switching the context quickly enough. Even in a block design, they had trouble to do this, to unsync certain things, for example, and switch to another condition. Like if you think about it, for example, if you try to switch from, let's say, feeling happy to feeling sad and back, I mean, then it's quite obvious that this is by no means trivial. And there, I think it might also help actually. So with the right paradigms and the right questions, you benefit. And that's another really interesting point or question there. Bigger one is the type of signal you feed back. For example, there you often fed back signals from regions of interest initially. Yeah. And you averaged across those. And if you choose them right, well, then they might be more compatible with the kind of changes you try to achieve. I mean, let me give an example. If you record signal directly from the motor cortex and you have localized it really well beforehand, then you really get the signal from that functional area that is relevant. So if you do then, for example, some type of motor training with a neurofeedback, maybe some motor imagery, then you get a very specific high SNR signal. If you do this with MEG, you still have this problem to disentangle it and so on. Same is true for deeper brain areas. So I think the spatial specificity also helps you to kind of give you a more precise signal in the end. And then last but not least is the work by Mitsuo Kawato, I think a lot of people will be aware of, is kind of, he was the first one to use these decoders in a real-time situation. So to now train subjects to actually achieve certain classification results, and even things they weren't aware of, so to say. There was this famous science paper he had on this topic, yes. the DECNEF. And I think, again, this shows there it's not a region of interest, but it's the relevant neuronal structure you train, because it's been trained by the classifier, which has been trained based on the relevant neuronal signals. So I think these kind of things help, despite having kind of the disadvantage of a slower signal, maybe less precise signal, So what you lose in terms of this temporal fidelity, I think gain in terms of fidelity in other domains.
So I think I think you've hit it on the head in terms of even not only looking at a region of interest, but also looking at, like you said, with multivariate classifiers, looking at the multivoxel pattern and you try to sort of change some aspect of that. And in one of your papers, you actually thought you showed that looking at connectivity as opposed to magnitude seems to be a better training signal as well. I mean, looking at the connectivity between regions, which is sort of a form of looking at multivariate signal as well. So, and that's interesting. Why necessarily maybe connectivity as it's assessed with MRI might be more. I mean, I think that this is exactly the edge of what we understand or don't understand. This is what's fun about doing this, is that you're exploring these signals and you're trying to figure out what is most relevant and what's most able to be used to train as well. So you also mentioned that the training effect. Let's say somebody is trying to use neurofeedback to reduce anxiety or something like that, or some people are trying to do post-traumatic stress, reducing amygdala. So you mentioned in one of your papers that the effect lasts about at least 20 minutes, but some people even argue. I mean, there were some early, I would even argue, maybe false starts. DeCharms had an early paper about pain mitigation. He said, he suggested that it lasted up to 6 months. What do you think of the efficacy and the length of time that this training effect? Maybe it accumulates over time if you keep on doing it, but I don't know. What's your insight on that?
I mean, you're really asking the big and the tough questions, I must say. But I think they are the crucial ones, to be honest. I mean, these are the really big ones, and I think they really go back. They go far beyond neurofeedback and the neurofeedback designs, and it was certainly something that has been puzzling us for a long time. And I think the story is, in my opinion, it's probably rather complicated. So first of all, short answer is yes, I believe that there are long-term effects and that they are lasting, and even on top of it, they might even get bigger over time. And now this is really puzzling. And there's some indications in some of the kind of clinical trials we had this big EU consortium network called BrainTrain, where people did kind of early clinical studies using real-time FMRI and neurofeedback. And they saw these kind of effects that, funnily, effect size seem to almost go up rather than go down, even though the training wasn't performed anymore, or the neurofeedback training wasn't performed anymore. And I think this has a lot to do with the fact that we don't understand plasticity and learning properly yet. But one thing we know is, for example, what matters is sleep. So how you, for example, structure your sessions, if you have them all on one day or on several days, and what gaps you leave in between, actually matters. So it's not only the dose. So there's a lot of talk about how much dose of neurofeedback do you need, but probably placing the dose strategically plays as big a role as the dose itself. So I think it really goes back to general training and learning paradigms, not only related to using now the bolt signal and your own brain signal for training and learning. And what makes it really hard, of course, to understand and study it is simply the fact that these are really extensive studies. I think sometimes people forget this, but when people talk about, for example, 8 sessions of neurofeedback, it often means they've actually put a single person 8 times for 1 hour into the scanner and did this experiment. And then you might have some results. And if you now try to work out all these parameters, you basically do this maybe 10 times. And then you have done 1 subject and you want to do your 20. It actually becomes a tremendously large study in the end. But these are really interesting questions. And maybe to add one more point to it is, I think what would be also fascinating is to combine it with other approaches. When you think about learning, so perhaps pharmacological interventions, perhaps also, there are beautiful brain stimulation techniques out there now. And I don't see them as mutually exclusive. Rather, maybe a nice, productive combination might actually give us the best, the quickest learning effects. Perhaps combining it with TDCS or something like that.
Yeah, no, and ultimately, hopefully, I think that it's close to being something. I always mention in terms of clinical applications of fMRI, this is something that could be a different type of thing where it's therapy that has real impact in that regard. I've seen nothing but mostly positive results. And so, and you're right, with multimodal integration, there's so much more that could be done. So yeah.
Maybe when you, if I may comment on that regarding therapies, I think that I would see 2 advantages. As far as we know now, it looks like, there are no, let's say, severe side effects of neurofeedback training. So I think that's one plus when you compare to other therapies, often in, let's say, in the neurological psychiatric domain. And the other thing, I think, which most volunteers comment on also patients, is this kind of feeling of self-efficacy. I think is quite important as well. It's you yourself who actually does or learns that. And I think these are 2 really positive aspects about the neurofeedback approach. One shouldn't forget about.
Yeah, I completely agree with that. And I think people quickly say, oh, it's just another form of biofeedback. Biofeedback really is just looking at, skin conductance or things like that as feedback, but this is actually looking at the brain processes. So it's has so much more potential for being targeted and effective. So great.
I fully agree.
Yeah. Well, I hope you at least continue working on that area to some degree. I think that your work has been really, really helpful to the field. So now to talk about things that you've been focusing on in the context of the talk that you gave, using MRI to probe microstructure. And so it seems that over the years, obviously, people have found that MRI is sensitive to, all kinds of things, white matter, gray matter, susceptibility, perfusion. And now with diffusion, you can look at fiber tracks and directionality. But I think that, and I think that what you're doing, and definitely correct me if I'm wrong, I think that, first of all, MRI could be more quantitative. And then secondly, there's an abundance of physiologic information at the cellular level that still is relatively untapped, or even not understood exactly how it affects the MR signal. Like John Gore recently gave a really nice Lauterbur lecture at ISMRM, sort of suggesting this, that we're using MRI for so many years and we still don't know exactly what causes T1. What are the physiologic and biophysical mechanisms? So what is, so I like how you framed your talk. I was privy to the slides, and so thank you for sending them. And so what would you say, so what is a cytoarchitectonic atlas, and what is known about myeloarchitecture?
So I guess we'll come back to this question about how quantitative MRI is and what you can extract from the signals. But what I wanted to say maybe just right away, I also believe that there's much more in the signal we can exploit, but we also need to understand, I think. And in terms of what can you potentially get out from MRI, I think what is really attractive for us is in the domain of neuroimaging and neuroscience is we can, for example, use it to revisit some of the older anatomy. So mainly, I call it maybe the macroscopic anatomy people have done, or mesoscopic one around 1990 up to 1920, 30s mainly, like Vogt and Vogt, Flechsig, Corbini and Brodmann, for example. I mean, a lot of people are aware of these atlases and that work. And I think we now really have the chance to get there in vivo, to get similar maps out. I mean, we are not there yet, to be very clear, but there's this chance. And I think if we push hard enough, we can probably get pretty close to that with MRI non-invasively. And then, of course, we have many more options. And why are we interested in those atlases? I think there are many reasons, but one important reason is that we also by now understand that these parcellations based on cytoarchitecture means what type of cells you have where in the cortex, and what are the densities, what are the cell sizes. I think the Big Brain project is a nice example for that. We know that those parcellations, they're also meaningful in terms of function. So you can distinguish, for example, primary motor cortex from primary somatosensory cortex based on their cytoarchitecture and based on their myeloarchitecture, which would be basically the fiber structure, the myelinated fiber structure in the cortex, where you also find a lot of myelinated fibers. And incidentally, actually, when you count the number of fibers, most of them are actually in the cortex. These are the short ones, not the long ones you have in the deep white matter. I think a lot of people are not aware of that. So they're really important for the connectivity of the brain. And so it's really interesting for parcellation. And in a sense, why is it important for function? Because it supports probably certain functional input or output better than other anatomical structures. And so in a sense, it even goes beyond just delineating and parcellating certain areas from others, but maybe also kind of understanding better how each area works when you have access to the myeloarchitecture and the cytoarchitecture precisely in vivo.
Yeah, it just occurred to me, one of the huge limits in high resolution fMRI, so we're trying to look at layer fMRI, and it's just simply that we don't, the idea is that you're based on sort of our electrophysiology literature, that upper layers maybe send output, lower layers have input, and that's just based on electrophysiology. But if right, I mean, it just occurred to me that if you could actually use myelo and cytoarchitecture information using MRI to characterize this, you immediately have the output input structure across the cortical sheath in that regard to inform the fMRI signals. So that would be really incredibly useful. That's just one example, but
and you could also present it like this. Ask yourself. Often we use literature information to inform modeling. This could be maybe neuronal mass models, like really formal, mathematical models. This could be maybe even cognitive models. So you go back to the literature. And what is typically the literature? If we are lucky, it's the mean of the population. Often it's the mean of a very, very special population we look at effectively. And then we take this and we kind of almost pretend as if this applies to your completely different group you have scanned now, for example, with functional MRI. But if you could take the same group and actually identify their individual properties,
Yes.
Wouldn't it be much more accurate, much better what you get out? I mean, this is a claim, but I would be quite optimistic that that's the case. If we wouldn't need to go back to the literature values, but we can actually do this in the individual in every study we perform.
Yes. Yeah, and not only for understanding individual variation, but of course, using clinically, you need to have that individual information to sort of assess the subtlety of some of the pathology. Yeah, so let's talk maybe a little bit about your approach. So starting out, the idea is that you have, MRI has many different weightings, and typically clinically you use T1 weighted, and tumors show up, and clinicians can interpret this. But when you really want to compare across scanners, and you actually want to get information that's more stable and maybe more relevant to what's actually going on, you need quantitative maps. So how do you go from your weightings to quantitation, and what do you end up quantitating then finally, as far as that's concerned? And not to go into, obviously there's many details in terms of how you synthesize how you do the calculations, but just generally.
Yeah. So I take kind of a bird's eye view on it. As you said, I mean, the quantitative MRI is a big field of research and has been actually for decades now, and people have been trying this for a long time. But I think what is special is now that we are at a time point where just the MRI scanners got so much better, the technique, so it makes the quantifying values also much easier. So what do we mean by that? I mean, typically when you look at, let's say, a T1 weighted image, you have this issue that, let's say, Peter, I would scan you at the NIH, and then I would bring you over to Leipzig, and we would scan you here on a 7T system. And then we would put the two images next to each other. And even if we ran the protocol with the same parameters, we would probably get different numbers out, so to say. So the numbers, the signal intensities are not really meaningful. The differences might be the contrast, but the numbers themselves not. And quantifying it is not so unusual when you think about normal science. I mean, standard science would always try to quantify. You always have a metric coming with that, an absolute value you would calibrate. So you would make sure that, for example, your thermometer gives you the same temperature independent of where you use it, for example, or if it's an electric one or a mercury thermometer, right? And so that's the fundamental idea. We want to get the same numbers out. And how do we do this? And that's also similar to other, let's say, areas of physics. Often it means calibration, something like that, some additional scan. So you might get your T1 weighted scan. Let's say we look at a T1 quantitative map versus a T1 weighted scan. Then a T1 weighted scan is usually one acquisition. A T1 map usually relies on, let's say, at least two acquisitions with slightly different parameters I've used, slightly different protocols, and maybe an additional reference scan. And then what I can do is I can kind of solve an equation to get the T1 out, and you solve for T1. And that is then independent of the exact protocol setup I have used for acquiring these two protocols. As long as I know the parameters of the protocols, I'm fine. I can estimate T1 from that. And that should be the same at different places. Of course, details matter, but the fundamental idea is that it's comparable or much better comparable across time points and sites.
Yes.
And in a sense, you could, maybe another viewpoint might be also, and that's a bit more geeky, in a sense, but when you go back and you think about what are the fundamentals, again the simple ones in MR physics, it's the so-called Bloch equations we use, and these phenomenological equations that describe the signal. And what you do is, in a sense, you get a lot of different signals, and then you fit the Bloch equations, and they have the parameters T1 in there, T2, for example, in there, and you get those out. So it becomes kind of an inversion or fitting problem in the end, the quantitative imaging. And typically we would extract these physical parameters like relaxation times or maybe magnetization transfer rates.
Yeah, and that's a wonderful overview. And the parameters you get out, right? You get like things like proton density. You get T1, T2, T2 star, megaspace transfer, as I mentioned, and from that, then you can then infer things like the amount of myelin, the fiber track directions, the amount of iron, which I'm still curious about, and we can maybe talk a little bit about that, separating blood iron from tissue iron. And actually, even more interestingly, I've seen a lot of work, including your own, sort of looking at using what's called like a G ratio to get at the size of the cells, or some people actually even Peter Basser here at the NIH is trying to look at neuron size, and who knows, we might be able to get neuronal density. I would love to have neural density maps potentially. I mean, more calibration potentially. But so as far as collecting this information, quantitating it, reduces the variability across scanners. Just before I go on with that, I just want to ask, so I noticed that, I mean, still there's variability, and it might just simply be in the signal to noise across scanners in terms of how you fit these equations. And so it doesn't eliminate it, it just reduces it substantially. That's, so with myelin mapping, just to put it in context, myelin is, and I've been learning a lot about myelin as far as how it's relevant. And I always thought myelin, was always there, and it's always just simply to make the neuron signal go faster. It's for, it's more efficient transfer. But it seems that, and you've been making myelin maps, starting with the myelin maps, and they're really fascinating, because the areas that show high myelination are, to me, counterintuitive. Of course, you have the myelin in the tracks, but then you have primary visual, primary motor that show this massive amount of myelination. So why is that? And how do you get the maps? And why do you think, how is the myelination different than what we understand? And maybe why is that the case?
Okay, all right. You might have to remind me of some of the questions. So I'll try to walk through it. So how do we get the maps? So I think in terms of understanding how it probably helps to think about it is, now we have these quantitative parameters, physical parameters, and what we try to extract from those are biological parameters now. And that's what we would call then the in vivo histology, for example, using MRI. So there's another crucial step here. It's going from kind of the simple model Bloch equations, which are true and nicely applicable in water, in a simple model system, into this really complex brain tissue where all kind of things are happening, multiple compartments. I mean, it's a real headache when you try to model it. So that's the big challenge. We try our best, and as a community, I should say this, a lot of people are active in this field. And so what we are trying to do is build parsimonious models of tissue to explain, then, for example, you might have different compartments. You might have heard about, let's say, this myelin water compartment, where you have water near myelin sheaths, for example, which has different characteristics because it's got a different mobility from the kind of free water that might be the extracellular water not close to any membranes. And there are many other models in there. You've mentioned G ratio, which relies on certain diffusion models. And so fundamentally, we build these models now. And we take the quantitative parameters, sometimes the weighted images, to estimate then these biological parameters. And again, it's this inversion problem. And you mentioned this briefly, that one aspect is that we really try to get as many different images and contrasts as possible, because that makes the inversion simpler. I mean, simply speaking, the more information you have about the underlying microstructure from different viewpoints helps you to estimate the microstructure. So that's kind of, again, bird's eye view on it, what we try to do. And we really try, ideally, our best models are the ones that work ab initio. So that means really there are no free parameters you fit, just natural constants in there. And the rest of it is physics, biophysics and you build the model. But that's really, really hard. So now coming to the myelin mapping. So there are different approaches towards that. The one we use relies very much on T1, measuring T1 and R1. And that's a very, very simplified model of the signal. But still, it works amazingly well, in particular in the cortex. There seems that there's a high correlation simply between the R1 value, so this quantitative measurement, and the myelination. And that's what we exploit. And why do we use this, let's say, simpler approach? Is simply because it's fast. And that means we can get very high resolution. And that means, we can actually look at the cortex and the interesting structures in the cortex, because with too big a voxel, you don't see anything in the cortex. So that's where we are coming from. And now the question I think you were asking is, why does it look like that, the myelo architecture? And that's, this, of course, one of the fundamental questions, right? In neuroscience, why like that? Why does it look like that? And I could cheat a little bit and say, well, we know that it's highly related to cytoarchitecture, and it's because the cytoarchitecture is different between those brain areas. Yeah. But that just shifts the question, right? But in a sense, what I'm trying to say, there are obviously different cell types. For example, if you think about the motor cortex, you have these giant bed cells for driving the motor output, basically. And so one could see that it's probably related to that you have certain cells and certain structures and also connectivity structures you need to support the function again, in my opinion. And that leads to different levels of myelination. And in the visual cortex, this might be, you have these collaterals that probably have to kind of effectively connect things. And maybe one comment, because you said you looked at myelin a lot and what the meaning. So it's certainly there to speed up or increase the conductance velocity, but I think there are so many other kind of uses of myelin in the brain, and probably one important one is stability of your system. What is myelinated doesn't change so quickly anymore. So if it's really kind of well learned, it's probably getting myelinated, and that makes it really an interesting plasticity marker, for example, or development marker.
Yes. I've read in the literature about how, myelination might change with learning, and certainly, but also at the same time, prefrontal cortex, which maybe intrinsically can't be too fixed, is less myelinated. But it also seems like myelination is, I've been talking to people that suggest that it's, in indication of cardiovascular health or just general health, and you also suggest it's in terms of aging, you have myelination decreases in a very distinct pattern in the brain as people get older as well. So and this is actually the wonderful thing about MRI is that you open up for the first time, we're able to sort of map this thing, myelination. And I was curious, that other people try to use the ratio of T1 to T2, and they all seem to converge pretty well. And people would argue that we're just looking at, water around myelin, but not myelin directly. But even when you compare it directly to the myelin maps that are obtained other ways, it's pretty close. It's really close. So have you been thinking about using just simply, I know you've done it with aging, but have you been trying to do, for instance, even in the context of real-time neurofeedback, trying to look at myelination changes? Certainly cortical changes have been observed. It's not clear whether that's wrapped into myelination. Have you been looking at, thinking about myelination as an indicator of plasticity?
Absolutely, yes, indeed, and also in the context of the neurofeedback experiments. So in some of them, we actually have started acquiring these kind of multi-parameter mapping techniques for mapping maybe myelination and also what is related is kind of iron concentration, and I think there's some evidence that there are actual anatomical changes due to the neurofeedback training as well, and in connected regions. That's kind of what we've seen, at least anecdotally, I would say, and not necessarily exactly the region you train, but it might be the one that is connected to it, which is quite interesting if you think about kind of motor basal cortical loops, for example, this motor loops. One thing I would really like to bring together and is this aspect of the microstructural imaging with training and the functional training, for example. So I think neurofeedback is a really interesting paradigm again at this point, because you can so selectively train something in the brain. So it's intrinsically quite well controlled.
Yeah, that's a really good point. That's a really good point, because usually when you're training, like for instance, I was always surprised with, early on people did a juggling task, and for some reason MT medial temporal areas showed changes, and nobody understood exactly why. But in terms of neurofeedback, you could actually say, we're going to measure these based on the feedback. You're going to change the activity, and then now you can directly look at myelination. Yeah, that's great. That's great.
Yep.
Maybe let's shift gears a little bit. Iron mapping. Iron mapping is also something that just sort of emerged a lot more in, especially with high field, which lends itself tremendously to doing sensitive iron mapping. And it's very clear that there's certain structures in the brain that have high iron deposits. But it seems that in your talk, you paint this picture of iron sort of being an indicator of, iron seems to correlate where there's myelin. Also, it's essential, but at the same time, too much iron is an indicator of disintegration. So do you want to maybe talk a little bit about iron in the brain? And how do you actually, and also the other question is, how does one, certainly iron is in the blood, that's a basis of bold contrast. How does one separate cleanly measurements of blood iron versus tissue iron?
Yeah, so I think that the story of iron in the brain is also a very complex one, and not well understood yet. I think one could say that. And there's certain, I think iron is so interesting because it's really got the two faces, basically. One, the positive, the good face, and the other one, the negative, the evil face, in the sense that you need it for, for example, when to produce myelin, how do you say, for enzymes, for example. So it's really something that is fundamental to the physiology, basically. Wouldn't work without iron. On the other hand, we also know that when you have these extreme high iron concentrations and deposits that are not physiologically regulated anymore, then you actually get the effect of that it basically builds up kind of radicals. So it really damages the tissue. So it's a very negative effect. So it's highly regulated in the body. That there isn't too much, and that it's in the right places and so on when everything works well. But when it doesn't work well anymore, then it gets dysregulated and then get all the trouble. And that's why it's interesting, both domains, kind of the natural healthy development, but also in the kind of negative, the poor aging and pathologies, for example, neurodegeneration. And there are a lot of neurodegenerative diseases that are somehow correlated, at least with iron, when you look into the literature. So it's really got several roles. And a lot of the iron is contained in ferritin, but you also have it, for example, in neuromelanin as another iron-containing pigment or substance, and so it kind of also lives in kind of different conditions, so to say, and it's important to distinguish those. And I think there's really much to learn, and I think there's when we get the modeling right, we can partially distinguish it. So we can really learn about these different iron states. And we also need to learn about them, because the simplistic modeling will not lead to really long-term promising results. I think we really need to understand it, what types of ions, what chemical condition it is in.
Yeah, and you mentioned, right, there's ways of, and right, you mentioned neuromelanin, which I just maybe in the last several years learned about. That it's a very it's another strong indicator of, I think, dopamine, or maybe I'm not.
That's right. Yeah, exactly. It is. The classic example is, and it's one we study as well, is that you find a lot of neuromelanin in the substantia nigra, and it's kind of, how would you say, what's the right word? It's part of the process of dopamine, kind of one of the products you get out, end products in the end. So you find it there, it accumulates there, and it scavenges iron. So it contains quite a lot of iron. So when you look at these dopaminergic neurons in the substantia nigra, they contain quite a lot of neuromelanin, highly concentrated, and highly concentrated iron there as well. And I think it's one of the contrasts that's quite well known in MRI there.
Okay. Yeah. So that's a unique application that could have all kinds of implications for addiction, to depression, to all kinds. Knows. I mean, so that's just at the very beginning of that. Maybe as I realize we're almost coming to the end of this, but there's so much to talk about. Obviously, if anyone's listening to this and this is before your talk, listen to your talk, or even check back on it at OHBM, since. I did have a question about, you make these beautiful maps of iron in the superficial white matter versus iron. You differentiate between iron in the deep white matter, or the sulci, versus superficial white matter. So what's the significance of the differences between these maps? And it seems like you tie it in with U fibers connecting visual areas, and it's like there's a really rich story here.
So this is a really interesting observation, and we and others have made this when you acquire, let's say, susceptibility sensitive images like T2 star weighted images or susceptibility maps. You see these hyper intense stripes just underneath the cortex, and it's exactly where you find the type of white matter, so-called superficial white matter, very thin stripe of about 500 micrometer thickness that connects, for example, it contains the U fibers that connect two adjacent gyri, basically. So they form this kind of U shape. And that's why they're called U fibers. And what is interesting is, so we were looking into this contrast much more closely because we didn't fully understand what's going on there. Why are they bright? Why do they look different from deep white matter? It's white matter. Shouldn't it look exactly the same? So you start wondering about it. And so we looked into it. And what's quite interesting is that exactly in that area, you find a very high iron concentration, much higher than in the deep white matter, and certainly much higher than in the gray matter, which is right next to it. You go from gray matter into this area with very high iron concentration, and it kind of goes down to the deep white matter again. And why we find it so important is because when you look at it from the angle of you're acquiring MRI data and you want to understand the tissue's microstructure better, then you want to have a unique marker. And this is kind of a unique marker because here the myelin, and I didn't say this, but the myelination estimates are the same in deep white matter and the superficial white matter. So now we can kind of distinguish it from deep white matter because it's the part of the white matter that's got the high iron concentration, much higher than the rest. Adding to that, what we could show is by modeling that this measurement, for example, of R2 star is highly related to the iron content in the superficial white matter. So it's kind of I would say it's an iron marker for superficial white matter iron. And what we could also see with very high resolution, 1 micrometer high resolution iron maps, we could see that the iron actually mainly lives in the somata of the oligodendrocytes and probably in the fibers. We are currently still looking at this very precisely with electron microscopy. But so that means now you have this link from MRI to iron, and you know where the iron is. So that makes it very likely that you actually have something like a fiber density marker, or maybe at least, a fiber maturity marker in a sense, because we also know that iron levels can change with the maturation of the fibers in there. And now you have a much better understanding, a much better interpretation of these maps, and you can use those in vivo and actually plot really beautiful kind of U fiber or superficial white matter maps. And we really understand very little about the U fibers in humans because they're so hard to image. When you look at them post-mortem, then you obviously can't do the functional experiment anymore. It's difficult to look at behavior. So doing these classic behavior structural relationship studies is really difficult without a non-invasive mapping technique. Wow. So we hope this will really help us to understand this important system in the brain, the superficial white matter.
So the U fibers actually seem to suggest that it's areas that show cortical areas that are adjacent and connected, and so sort of understanding how they're connected in that way. Yep. It's amazing also. So it's like we fill our gaps in our knowledge just with sort of blind spots, but then with MRI you sort of show that U fibers, and suddenly everyone's like, wait, yeah, that's right. We don't really have a deep understanding of this. We have to reconsider this. So it's a wonderful sort of iteration that happens, that's sort of spurred on by just these empirical observations that come through deeper and deeper use of MRI, which is wonderful. So just to there's so much more we could talk about, but just to finish up, there are many areas that you're studying. I mean, you're studying. I wanted to talk about a paper with aging, learning, Parkinson's disease, all kinds of things. What would you suggest for MR physicists or neuroscientists with some technical bent? What would you suggest would be the most interesting projects? What are the directions that have the most potential benefit in the future with MRI, at least, or multimodal?
That's a hard question.
Yeah, it's an open-ended question.
I think in my opinion, really great potential is all the different approaches that really try to approach questions more quantitatively. So this is now very abstract, but let me explain what I mean with this. This could mean, for example, what we discussed right now, to not just do some, shape analysis, and then you don't really know exactly what changes, but do the shape analysis, do the microstructural measurements, all of that, and put it together and try to quantify this as much as possible, because that gives you, I think, in my opinion, the strongest predictions, which can also most easily fail. And I think that's strong science, which you can disprove really clearly and easily, and maybe also with other techniques that are not MRI, because you've now given other people a biological measurement result and prediction, and they can go at it and critique it and so on. And similarly on the functional side, for example, I'm quite interested in these more formal models like neuronal mass models, where you get stricter and more clear predictions, which you might then be able to probe with electrophysiology, completely different method, from MRI. So if you ask me at this kind of more abstract level, I would say, yes, models, approaches that are really trying to put clear numbers to your predictions of what you are saying. And I said it's a bit of my taste, and it certainly, I think it's strongly formed by coming from physics, where you're so used to this, to make these quantitative predictions and then trying to tackle or disprove these models. And for me, of course, I'm completely excited about this possibility to look at microstructure now more and more quantitatively and trying to understand that better. And there are I couldn't even start. I wouldn't even know where to begin, what you can do with that. Actually, there's so many possibilities in terms of studies.
Right. And so related to this question, are there any, like for instance, when you compare histology, when you look at a histologic sample, are there any aspects of the histology that still remain not understood that MRI might lend insight into? Are there any connections to be made?
Yeah, absolutely. And when you think about the classic histology, I mean, there's really this big issue that it's you have all the vagaries of staining. It comes out differently depending on maybe who did it, what lab technician worked on, and all these kind of things. And MRI doesn't have that, right? When it works, it really gives you those quantitative values, and you can compare them. So I think that's a big advantage. It's three-dimensional. So I think with histology, you often really struggle with the fact that most of them it's 2D and it's slice-based, so the connectivities become really difficult to look at. So there's actually quite a lot of things we could potentially do even better with MRI than with histology, especially, I mean, when you think about the postmortem situation where MRI is much easier to perform than in the in vivo situation, because you don't have movement and so on. You can look at smaller specimens. So I think there's a lot of potential, and I think the techniques can help each other. So, for example, we've been looking into, for example, myelin stains, and we all know they look very different depending on whether you use Laxal's fast blue or you use NBP and so on. And MRI looks also very different, whether you look at T1, at proton density and so on. And I think they can really help each other, so you can understand each contrast better and each component. And we are quite excited about, for example, looking at lipids in myelin as one of the contrast drivers in MRI. And maybe they can help us to explain a little bit better where this T1 contrast, for example, comes from, and why it looks like that, and why it looks different, for example, in different brain areas. So yeah, there's a lot to do.
Yeah, no, that's exciting. That's a great answer, because it really does paint this picture that there really are more parameters to explore with MRI, and there's in other areas that it's used for, histology or whatever is not a perfect science, and MRI has a potential for not only improving that, but sort of coming up with new insights about things. So this is great. Well, I think actually we're at the end of our time, but I really do appreciate you taking the time to come on and chat and listen to my questions. But and definitely your work over the years has been pioneering with not only with MRI, not only with functional MRI, with real time. But now this whole field, I get this real sense that the direction that MRI is taking in this regard is extremely rich, and there's tons of opportunity. So I think you're leading the way. So I recommend everyone listen to your talk, and thanks for coming on.
Thank you very much, Peter. I mean, it was a real pleasure and a lot of fun to talk to you. Thanks a lot.